Hello, everyone. My name is Rafael dos Santos Peixoto. I'm a PhD student in Gene Fan's lab, and today I'll present my work about characterizing cell-type spatial relationships across length scales. So first, among the many analyses you can do with spatial omics, one of them is identifying cell types. And on the picture on the left, you can see a spatial transcriptomics data set of the mouse embryo. So we collected the gene expression information, we identified the cell types, and on the right you can see a proteomics data set where we capture protein information, identify the cell types, and in both figures, each dot represents a cell, each color represents a cell type. And as you can see for the picture, the cells are not randomly placed in the tissue, they are organized. And understanding this organization is very important, and this was the reason that motivated the development of the package. On the left case, in the mouse embryo, understanding which cell types are close to each other and which cell types are separated can be a good indicative of what these cell types will do after development. And on the right picture, on the spleen, understanding which cell types are next to each other might be relevant for understanding what are the functional tissue regions or what cell types are involved in the functional tissue regions of this organ. Besides understanding the organization of individual sample, it's also important to compare multiple samples. In this case that you see here, you have two human thymus samples, one from 11-year-old, the other one from 21-year-old. And when you see, just looking at these pictures, you see that the one on the left has a lot more cell types, a lot more cells, and the one on the right does not. So understanding which cell type relationships are preserved as we age might be a good indicator of what the relevant functions that these cell types are doing as they are captured, or they're still maintained as we age while the other cell types are not preserved. Additionally, it's also important to understand which scale this relationship happens. If you look on the picture now, again, each cell is a dot, each color is a cell type. You might not see any clear relationship from the red cells with the other cells, with the blue cell, for example. But as you zoom out more, you see that the red cells are surrounding the light blue cells. And if you zoom out even more, you start to see more of the relationships. So what I want to get from here is that understanding cell type relationships is important. It's also important to compare the cell type relationships. And it's also important to understand which scale this cell type relationship happens. So now, to explain our method, first, for a specific cell type of reference, in this case, we're looking at cell type B, we'll choose a neighborhood distance, and we'll draw a circle around the cells of this specific cell type, of this chosen cell type. Then, we repeat that for all the cells in the cell type, creating multiple circles. We unionize the circles, creating a neighborhood. And with this neighborhood that's highlighted in yellow, we calculate the proportion of all other cell types inside the neighborhood. Now we have original proportion of cell types inside the neighborhood. Our next step is looking at a particular length scale. We'll divide the tissue into grids, and the size of the squares in the grids are the chosen length scale. Now, we'll shuffle the labels inside the squares of each grid. We're not shuffling the positions of the cells, we're just shuffling the labels. And using the same neighborhood as before, we'll calculate a new proportion. So now we are obtaining what will be the new background. What is the proportion if the cells were randomly placed? And with this information, we'll have another proportional value for each cell type. We'll have our hypothesis, and we use a z-score to verify this hypothesis. So in summary, you calculate a first proportion, we shuffle the data at a particular length scale, get a second proportion, and compare these two using a z-score. We repeat this process for every cell type pair. So we have a z-score for every cell type pair, and we do this by creating grids of multiple length scales, so we get a z-score for multiple length scales. The results you can see here in the simulated data. As you repeat this process for multiple length scales, as you can see in the trend plot, we have reference cell type C and neighbor cell type B. We have a z-score value for each length scale we calculated, and we also have a significance threshold, which is a p-value of 0.05. corrected for Bonferroni for the multiple cell type relationships we have. And we determine the relationship as the scale in which the trend crosses the significance threshold. So for example, for a reference cell type B and C, we see that relationship significance are around 250 microns. But as we look for cell type A and B, A is the red cell, so they're still next to the B cell, but not as next as the reference cell type C. This relationship happens at a greater scale, which means that you need to zoom out more to see the relationship happening. And since we have multiple cell types to test, we create a summary visualization, which is the dot plot shown on the right. The color represents the z-score value, so red is a positive z-score, blue is a negative z-score, red means a colocalization, blue means a separation of these two cell types, and the size of the dot represents the scale this relationship is happening. So bigger dots represent a finer scale, smaller dots represent a greater scale. And we see this working on simulated data, but what matters is real data. So we move to the mouse cerebellum. The mouse cerebellum is a very structured organ, it has layers, and we're expecting to see these relationships being replicated in our analysis. And this is actually what we see. So we have Berman glia and Purkinje cells that are next to each other because they are within the same cerebellum layer. And we can see they colocalize in our method, as you see in the red dots with the stars. But they are separated from the oligodendrocytes, which are in a different cerebellum layer. And we can also see that when we apply crawdad to this data set. When we are comparing crawdad to other methods, we see that now we're looking at the Purkinje cells as the reference. We see the Berman glia are separated because they have a positive z-score. The oligodendrocytes are separated because they have a negative z-score. Berman glia is colocalized because of the positive z-score. But this distinction is not as clear in the other methods. And this is essentially because while crawdad preserves the same neighborhood and changes the background, the other methods preserve the same background but change the neighborhood sizes. So the results are different, as you would expect. Moving to a less structured organ than the cerebellum, we analyzed the human breast cancer data set. And even with less structure, less organization than the cerebellum, we still identified 4 groups: a group of immune cells, a group of one cancer type, and a group of another cell type, another cancer cell type. Looking into the multiple sample analysis, we analyzed 9 mouse brain data sets, 3 replicates from 3 different locations, and after calculating the cell type relationships for each cell type pair in each of the samples, we compared them and we applied PCA analysis, principal component analysis to these data sets. And when we looked into the reduced dimension space, we saw that samples that came from the same location were close to each other, meaning they have a similar organization than samples that came from different locations, as you would expect. And we're also able to identify which cell type relationships vary the most across locations and which ones were kept the same across replicates. We also analyzed the human spleen, as I presented in the first figure. We identified the 2 tissue units of the human spleen, the red and the white pulp. And when we looked into these particular relationships and their variance, we saw that some of the relationships were patient-specific. In this case, we had 6 samples, 2 samples from 3 different patients. And the samples, some of the samples that came from the same patient had the same relationship, but it also had effects of sample-specific. As you can see, even samples that came from the same patient had different relationships. In this particular figure, each color represents a patient, each trend represents a sample. And this, all I spoke before, is published, but now we are trying to look into region-specific analysis. So instead of just identifying relationships that are particular to a patient, we're trying to identify relationships that are particular to a region or a neighborhood in this tissue. In conclusion, crawdad is an open source R package that serves for analyzing cell type spatial relationships at multiple length scales. It allows a multiple sample analysis, so you can not only look at one specific sample, but you can compare multiple of them, as in the paper showed we compare 9. It's applicable to any spatial omics technology, either transcriptomics or proteomics, in which the cell types and the positions available. And I would like to thank everyone who made this work possible. Also, like to thank the organization for the opportunity and everyone else for paying attention to my presentation. Here's the QR code of the paper. And if you'd like to get free stickers, find me later. Thank you.
Questions?
Hi, very nice talk, very visual. So I have a question. For many data sets, we have some data coming in Visium and some, for instance, comes with Xenium. Obviously, you cannot compare them on the very close length scale because Visium would not allow that. But is it possible to use Crowbar and starting from a certain distance to compare these distance distributions for across samples, regardless of the technologies they've been profiled with?
I think I'll divide your question into two parts. The first part is talking about Visium. As Visium, we know, captures a large region of the tissue, which can happen like 7 to 10 cells within the same spot. We have not analyzed it as in this way because in our process of shuffling labels, if you have like 10 cells within the same location, it's hard to shuffle those labels. So we have not applied crawdad to Visium data. But if you're looking for comparing different technologies, let's say Xenium or Mirrorfish, I think it is possible as long as you take in consideration the batch effects that can come from each technology when you're identifying the cell types.
All right, thank you.
Thank you.
Thank you again, Rafael.
Thank you.
And the next we have talk